Very good morning, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to resume the chapter 14, that is respiration in plants. In the previous lecture, we have already covered the glycolysis part, and today we are going to see the next topic of this chapter, that is the citric acid cycle. If you have any doubts related to the previous lectures, you can check my YouTube channel for it. So let's start with today's lecture. So in glycolysis, right, we have seen several steps, right? So in glycolysis, normally two molecules of ATP are consumed during the double phosphorylation of glucose to fructose 1,6 biphosphate, right? So here, when the glycolysis is taking place at that time, two ATP molecules, they are being utilized, right? Or they are being used. Two molecules, same way the two molecules of NADPH2, that is nicotinamide adenine dinucleotide phosphate, are formed at the time of oxidation of glyceraldehyde 3 phosphate to 1,3 biphosphoglycerate, right? Each NADH is equivalent to 3 ATP, so that net gain in glycolysis is 8 ATP. So normally over here, when two molecules of ATP are consumed for conversion of glucose to fructose 1,6 biphosphate, right? So two ATP, two molecules of ATP are consumed. Same way, when the two molecules of NADPH2 is formed, right, upon the oxidation of the glyceraldehyde 3 phosphate to 1,3 biphosphoglycerate. So at this time also two molecules of NADPH2 are oxidized, and for this reason each NADH is equivalent to 3 ATP. So two molecules, see, if normally if each NADH2 is equivalent to 3 ATP, that comes to two molecules of NADH2, that two molecules of NADH, that is equals to 6 ATP, and already over here in two molecules are already consumed. So for this reason the total net gain of the ATP in the glycolysis is totally 8 ATP, right? So here for each NADPH 3 ATP is equivalent. So for two molecules of NADH, 6 ATP will be used. And the net gain, that is the two molecules of ATP were already consumed during the conversion of the glucose to fructose 1,6 biphosphate. So at that time, that is the 6 plus this two molecules of ATP, that comes to total gain will be the 8 ATP. That is the net gain in glycolysis will be 8 ATP, and this can be asked in MCQ also. Now, after this, the pyruvic acid, which is the product of glycolysis, right? This pyruvic acid is the key product, and this key product is going to be further breakdown of the pyruvic acid takes place and depends upon the need of the cell, right? So further, this pyruvic acid, it has the ability to go in different pathways, right? So it is based upon the need of the cell that this pyruvic acid is entering into which cycle. Now in normally in animal cells like the muscles, during exercise, when oxygen is insufficient for aerobic respiration, this pyruvic acid is reduced to lactic acid by an enzyme lactate dehydrogenase due to reduction by NADH2. So here in animal cells, like when the particular animal is carrying out any of the exercise, right? So the muscles, right? So the muscles are being contracted and relaxed. So at that moment, when the oxygen is in insufficient amount, right? At that time, what happens? This pyruvic acid, it is being reduced to a acid which is known as lactic acid with the help of an enzyme which is known as lactate dehydrogenase. And here the reduction takes place of the NADH2, and due to this, what happens? The pyruvic acid gets converted to the lactic acid. So over here you can see is this glucose gets converted to glyceraldehyde 3 phosphate, which you have already seen in the glycolysis. This glyceraldehyde 3 phosphate, upon the reduction of NAD to NADH plus H plus, that means it releases out the H plus, it forms 3-phosphoglyceric acid, right? So when it forms a 3-phosphoglyceric acid, here what happens? This 3-phosphoglyceric acid, it gets converted to phosphoenol pyruvic acid. This phosphoenol pyruvic acid gets converted to pyruvic acid, which we have till here we have already seen in the glycolysis part, right? So when this pyruvic acid is formed, this can be either this can either going for the fermentation part, or either it is going to form the lactic acid upon the reduction of the NADH plus from the NADH2, it is going to reduce to NAD plus, right? So here there will be the removal of the H plus ions, and for this reason this pyruvic acid is converted into the lactic acid. Next, this pyruvic acid, it is also going for the oxidation part as well as, right? So here what happens, this NADH, again the reduction takes place, and for this reason this NADH, and here also there will be the removal of the H plus will be taking place, and for this reason what happens, this NADH2 gets converted to NAD, right? Where this pyruvic acid gets converted into ethanol, and also it releases out the CO2. So here in the fermentation by yeast, the pyruvic acid is converted to ethanol and CO2. So yeast is, that is a Saccharomyces cerevisiae. So when this yeast is carrying out in the fermentation part, at that time what happens? The pyruvic acid is converted into the ethanol as well as CO2. And the enzyme which is involved in is the decarboxylase and the alcohol dehydrogenase, right? So these are the two enzymes which are responsible for the conversion of the pyruvic acid into the ethanol and the CO2. Now, in both the lactic acid fermentation and the alcohol fermentation, as you can see over here, first fermentation which is taking place, it is the lactic acid fermentation, and second fermentation, that is the ethanol plus CO2, like that is simply said as the alcohol fermentation, right? This two of the fermentation, this in both, that is the lactic acid fermentation and the alcohol fermentation, very less amount of energy is released. So when this two of the fermentation is taking place, very less amount of energy releases observed. Now this yeasts poison themselves to death if concentration of alcohol reaches above 13%. So if more amount of alcoholic fermentation is taking place, and if that alcohol percent reaches to above 13%, then the yeast which are present inside it, which is responsible for carrying out the fermentation, that yeast die, or the death of those yeast occurs by themselves only, because the alcohol percent is reaching more than 13%. Now the final product of the glycolysis, that is pyruvate, which is simply said to be the pyruvic acid, it is being transported from the cytoplasm into the mitochondria for further breakdown. So the pyruvic acid which is being produced upon getting or upon carrying out the glycolysis, this pyruvic acid transfers from the cytoplasm into the mitochondria. So now this pyruvic acid is entering inside the mitochondria. Now the oxidation of pyruvate to acetyl-CoA is done to produce CO2 and NADH, right? So simply when the oxidation, that is the loss of the electrons which is taking place, it occurs in the pyruvic acid. So this pyruvic acid, right? This pyruvic acid and the oxidation which is taking place of the acetyl-CoA, right? This is done, right? So the oxidation of the pyruvate to acetyl CoA is done, and upon carrying out or upon occurrence of the oxidation part or oxidation reaction, the production of CO2, that is the carbon dioxide, and NADH is observed. So the oxidation of pyruvate to acetyl CoA is responsible for the production of the CO2 and NADH. So you can see here the pyruvic acid gets converted to acetyl-CoA with the release of NADH as well as the CO2. There is a special enzyme to catalyze this reaction, and that is pyruvate dehydrogenase, as well as a coenzyme is being used, right? That is the NAD plus, where you can see over here, right? As well as the cofactors are also being used, that is the Mg2+. This acetyl-CoA enters a cyclic pathway which is known as TCA cycle, or simply it is also known as Krebs cycle. So upon the conversion or upon carrying out the fermentation part, right? So first the glycolysis took place, and the end of the glycolysis, that is whatever product of the glycolysis has been formed, that is pyruvic acid. This pyruvic acid is responsible for carrying out or for entering inside the two types of fermentation. The first type of fermentation is the lactic acid fermentation, and second is the uh, alcohol fermentation, right? So this pyruvic acid, in the presence of the pyruvate dehydrogenase along with the cofactor Mg2 plus as well as several coenzymes, that is NAD plus, the pyruvic acid undergoes the oxidation and it converts to acetyl CoA with a release of NADH plus CO2. So when this acetyl CoA is being formed upon the oxidation of the pyruvic acid, this acetyl CoA is entering into a cyclic pathway which is known as TCA cycle. And simply, this TCA cycle, the full form of TCA cycle is tricarboxylic acid cycle. This this TCA cycle is also known as Krebs cycle. So now we are going to see about the next type of the cycle, and that is the Krebs cycle, which is also known as tricarboxylic acid cycle. This TCA cycle was first discovered by Hans Krebs in 1940, and for this reason, this cycle is also known as Krebs cycle. This cycle is also known as TCA cycle because the initial product, that is the first product which you can see over here where I am showing the cursor, as it is producing the citric acid. For this reason, it is known as citric acid cycle also. So there are three different names of this cycle. The first is TCA cycle, second is Krebs cycle, and third is the citric acid cycle. So let's see the citric acid cycle. So now whatever pyruvate was there, that was a 3-carbon compound, right? So this 3-carbon compound, in the presence of the acetyl CoA enzyme, it is going to form the acetyl CoA, which is going to be a 2-carbon compound, because here there was the oxidation which was taking place with a removal of a phosphate, with the removal of 1 carbon dioxide, right? So for this reason, this 3-carbon gets converted to the 2-carbon compound, which is simply known as acetyl CoA or acetyl coenzyme A. Now this acetyl coenzyme A with a 2-carbon compound, right? This enters inside the citric acid cycle. Firstly, it forms an unstable compound, but after that, this 2-carbon compound, it combines with the oxaloacetic acid, that is a 4-carbon compound. So the 4-carbon compound plus the 2-carbon compound acetyl CoA, these two of them combine with each other to form a stable compound which is known as citric acid, and this is a 6-carbon compound, and this is quite stable in nature. So for first step involves the addition of the oxaloacetic acid with the acetyl CoA. So the oxaloacetic acid, it is a 4-carbon compound. It binds with the 2-carbon compound, that is acetyl CoA. So when the combination or when the addition of this 2-carbon takes place, so 4 plus 2, that comes to 6-carbon, and it is going to form an acid which is known as citric acid. This is the first stable product which is formed, and for this reason this cycle is also known as citric acid cycle. Now this citric acid, which is a 6-carbon compound, is now going to be reduced, right? And we need to reduce it. So here 1 carbon, that is 1 molecule of carbon dioxide, it is released out, and along with that the NAD is getting converted to NADH plus H plus. So here the conversion of NAD occurs, and that NAD gets converted to NADH2, right? So this citric acid, 1 carbon is released out in the form of CO2, and here the NAD gets converted to NADH2. So upon the release of 1 carbon, this citric acid is converted into a 5-carbon compound which is known as alpha ketoglutaric acid. So citric acid, which was a 6-carbon compound, upon the removal of the CO2, it gets converted to a 5-carbon compound which is known as alpha ketoglutaric acid. Now this alpha ketoglutaric acid, again 1 carbon over here is released in the form of CO2. Again there will be the Again there will be the conversion of NAD is observed over here. The NAD gets converted to NADH2, and here the ADP, that is the not the here not the utilizing of energy is taking place, but here the energy is forming. For this reason, the adenosine diphosphate, that is the ADP, gets converted to ATP, that is adenosine triphosphate. Now, as this was a 5 carbon compound, but from this 5 carbon compound, again one molecule of carbon is been released out. For this reason, this 5 carbon compound is converted into a 4 carbon compound, which is known as succinic acid. This succinic acid, which is a 4 carbon compound, is converting itself to a malic acid, which is again a 4 carbon compound. But here the reduction is observed, which is the FAD gets converted to FADH2. So it is also going to do the same thing, just like the NAD gets converted to NADH plus. So here the FAD gets converted to FADH2. So for this reason, the succinic acid gets converted to a stable another 4 carbon compound, which is known as malic acid. This malic acid again gets converted to a 4 carbon compound, which is oxaloacetic acid, but here again the NAD gets converted to the NADH2. So here when the oxaloacetic acid is formed, again the second cycle is running. So again this acetyl CoA, which is a 2 carbon compound, combines with the oxaloacetic acid, which is a 4 carbon compound. Again it forms citric acid, then alpha ketoglutarate acid, and again the whole cycle continues. So as this citric cycle, this TCA cycle, what it says is about that with the help of the pyruvic acid, which was the final product of the glycolysis, with the help of the enzymatic reactions, it is forming the acetyl CoA, and when it binds with the oxaloacetic acid, it forms the stable compounds, and further it reduces, and upon the reduction it again forms the oxaloacetic acid, which again combines with the same acetyl CoA to continue with the another cycles, right? So here you can see that acetyl CoA combined with oxaloacetic acid and water to yield citric acid in presence of enzyme citrate synthase to release CoA. So citrate synthase is the first enzyme which is responsible for binding of the oxaloacetic acid to the acetyl CoA, and the citrate synthase is responsible for releasing out the coenzyme A. Next is citrate is then isomerized to isocitrate, and it is followed by two successive steps of decarboxylation. So you can see over here decarboxylation, D means the removal, carboxylation, that is the carbon dioxide. So as you can see, there is a removal of the carbon dioxide is observed as two carbon dioxide molecules are removed. For this reason, it is said to be over here that it is followed by the two successive steps of decarboxylation. So two times the decarboxylation is taking place, and this leads to the formation of the alpha ketoglutarate acid, and further it forms a succinyl CoA. In the remaining steps, the succinyl CoA, that is the succinic acid, is oxidized to oxaloacetic acid, allowing the cycle to continue. Over here, there are 3 points in the cycle where NAD is reduced to NADH2. See, you can see over here where I'm showing the cursor. The first is the conversion where the citric acid gets converted to alpha ketoglutarate acid. There is a first reduction reaction which is observed. The second is over here, that is the conversion of alpha ketoglutarate acid to succinic acid. And the third is from the malic to oxaloacetic acid. So three times the reduction of the NAD plus is taking place to NADH2. And at one point, there is reduction of FAD is observed to FADH2, that is through from the succinic acid to the malic acid. You can see over here the reduction of FAD is taking place, and it is reduced to FADH2. Now, a molecule of glucose produces two molecules of NADH2. So now I would like to tell you that please you should have proper attention over here, because this is quite confusing. So here now you understand one molecule of glucose, it produces two molecules of NADH2, right? So one molecule of glucose, it is responsible for producing 2 molecules of NADH2. So 2 ATP and 2 pyruvate while undergoing glycolysis. So the glucose, it produces 2 molecules of NADH2, 2 ATP, and the 2 pyruvate, that is 2 pyruvic acid. These three things has been produced by the glucose molecule. Now the 2 molecules of pyruvate are completely degraded in Krebs cycle to form 2 molecules of ATP. So here you can see over here that, see here, the ATP is been generated. For this reason, it is written that the 2 molecules of pyruvate, it is completely degraded and they are forming 2 molecules of ATP, as well as 8 NADH2 as well as 2 FADH2. Now you can see over here, as uh, as two cycles are running, that means one molecule of pyruvate, that is 3 carbon, another molecule of pyruvate, that is 3 carbon, as two of them are running side by side. So from, if one is is getting reduced to NADH2, so 1, 2, and 3, that comes to 6. So here you can see, but over here you can also see that this NADH plus H, right? So this is also taking place over here when the conversion of the pyruvate to acetyl coenzyme is taking place. So what happens over here? It is going to form 2 molecules of the ATP. So 2 molecules of ATP, because one ATP is here and second ATP is from the another second pyruvate cycle which is taking place. So 2 ATPs are formed, 8 NADH2 are formed. How 8 NADH H2, 1 is over here, second, third, and four. 4 into 2, that comes to 8. And the third, 2 FADH2, because only one conversion over here is observed. So one for this cycle, one for the second cycle, that comes to 2 FADH2, right? So this way, in the Krebs cycle, the 2 molecules of ATP, 8 molecules of NADH2, and the 2 molecules of FADH2 is formed, right? So this is the reaction of the Krebs cycle which is taking place. That is the pyruvic acid plus the 4-NAD plus FAD plus 2H2O plus ADP plus phosphate. This, in the presence of the mitochondrial matrix, gets converted to 3CO2, that comes to the release of the carbon dioxide, the 3 molecules of the carbon dioxide, as well as it is responsible for the release of 4-NAD H as well as FADH2, plus it is also removing the 4H, that is the protons, as well as 2 molecules of ATP. So this way the whole Krebs cycle is running for the 2 different molecules of the pyruvic acid. I hope you are very clear with today's topic and I hope to see you again. Thank you.